Donc, Miguel, tu peux y aller.
Parfait. Bonjour à tout le monde et bienvenue à la présentation du projet de thèse de l'étudiant Ruben Leroy. Il va nous parler aujourd'hui sur une évaluation des impacts de différents traitements de coupe partielle sur la biodiversité. Ruben, tu as approximativement entre 30 et 35 minutes pour nous parler de ton projet. Et par la suite, on fera deux tours de questions. Premièrement, on va faire des questions du public et par la suite, à port fermé, on fera deux tours de questions avec le comité d'encadrement et l'équipe de direction. Mais avant tout ça, je voudrais vous dire que Ruben va faire sa présentation en anglais. Alors, les personnes francophones, on s'excuse. Mais Ruben, tu as la parole. Tu peux commencer quand tu veux.
Yes.
You can start, Ruben.
Okay, thank you. Well, welcome everyone. My name is Ruben Leroy, and I will be presenting my PhD proposal, which is evaluating the impact of modern partial harvest methods on biodiversity in boreal forests, which is directed by Miguel Montoro Girona and co-directed by Gabriel Pigeon. So let me head straight into it. Yes, yeah. Okay, forest biodiversity is decreasing worldwide over the most recent decades and keeps declining under its current situation. Now, of all the biomes, the boreal forest biome encompasses one of the largest biomes on the planet, and with nearly 2/3 of this boreal biome being falling under some form of management, a decline or destabilization of tree diversity, forest capital, and forest biodiversity has been concurrently observed. This is due to clear-cut being the most used silvicultural practice for coniferous forests, causing the forest to obtain unsustainable damages in the biotic and abiotic traits over all the forest layers, whose impacts are normally not seen under natural disturbances. This is because in clear-cut, the removal of the majority of the standing trees in one operation is considered as one of the most intensive disturbance regimes which can happen to a forest. The implication of the practice directly adjusts the environment, such as elevated temperatures, unfavorable soil, weakened resilience, and forces the stand in favor of specific species groups, such as pioneers and invasive species, causing the eventual imbalances on the stand. However, clear-cut practices can be a highly efficient tool in healthy environments that are able to withstand the practices. When this is not the case, though, the intensive disturbance rates of clear-cuts can create harmful impacts to the habitat, resulting in various forms of habitat loss. The current continuation of these management practices lead to various forms of habitat loss, ecologically unstable environments, and reduced diversity and abundance for the inhabiting communities, endangering the fundamental ecosystem services that the boreal forest systems provide. Now, in Canada, clear-cut also remains the most dominant forest management form up until now. Yet, through new knowledge, initiative, and ecosystem concern, new alternative practices were established which focus more on the ecological processes and strive for biodiversity maintenance at their core. The ecological concern on clear-cut impacts drove the Canadian forests to establish new projects, such as the MISA project in Black Spruce Boreal Forest of Quebec, Northern America. Foregoing research already yielded thorough knowledge on the growth, regeneration, and mortality traits of these forest practices, yet gaps about the inhabiting communities remain present, providing the study with the opportunity to research further into the subject. Bioindication carries similar gaps due to the inconsistency of evaluation and inaccuracies caused by the limitational traits between the taxa. In response, this study tries to tackle these dilemmas with new methods. So, in ecosystem-based management, one strives for sustainable forest management through the adjustment of resources and reduced differentiation between natural and human-made disturbances. But what does this mean? Well, foregoing research discovered that natural disturbances drive the dynamics of the structure and functionality in forest ecosystems. Yet, these disturbances markedly differ from the human-made disturbances we administer to the forest when harvesting. Since discovering the value of disturbances in forests, research have focused on creating designs that most effectively incorporate or mimic natural disturbances, such as explained in the ACO model, as you can see on the figures, or in natural disturbance-based management, natural disturbance simulation concepts, etc., which range over various traits such as frequency, patterns, intensity, spatial extents, etc. These concepts combined resulted in the creation of the ecosystem-based management. Now, partial harvest treatments are part of this management due to several reasons. as partial harvests spread the harvesting over 2 sessions and leaves some of the parts of the habitat residual, they reduce the intensity to more intermediate levels. Its harvest traits, such as spatial patterns, gap sizes, age classes, woody debris retention, and much more, are flexible to the harvesting approach, which makes them highly adaptable to the context of each environment. Finally, the retention of the residual combined with the woody debris provides fundamental conservation traits which aid the ecological condition of the forest. Research on the effects of partial harvest not only found reduced impacts of the implied harvest, but also found indications that improved the conditions compared to unmanaged forests who rarely experience disturbances. So, the extensive advantages of partial harvest give high indications that these harvesting methods provide a more beneficial approach compared to the traditional harvesting approach that still dominates the Canadian forests. However, since various aspects of the partial harvest remain scarcely researched, the uncertainty remains if the benefits of the harvest method actually apply to all the various forest habitats of Canada. Now, in my study, I concern myself on the various harvesting impacts on the inhabiting faunal and floral communities. Since we know that the traditional forest management, clear-cut, does not always include sustainable forest management, and biodiversity is reaching a concerning decrease rate, the need to find alternatives is at its climax. Yet, inconsistency remains on how to represent the ecological condition of forest habitats. To approach this, we refer to 3 common communities present inside the boreal forest, which are often also selected as representatives for the identification of the ecological condition of the forest. First off, plants. The understory vegetation responds to a wide variety of environmental conditions, such as light intensity, soil temperature, soil microorganisms, soil compaction, etc., which makes them very useful bioindicators for monitoring the changes in a local condition. On top of that, their presence is essential for the functionality of ecosystem services, making them a fundamental ecosystem regulator. Under clear-cut management, this community showed major compositional and structural changes, while retention practices, such as partial harvest, reduced the impact significantly to levels where maintenance of the composition and structure remained possible. Insects, on the other hand, are the most diverse animal group on the planet. Their role is mainly visible in the maintenance of services and diversity within the ecosystem, such as decomposers, food web interactors, nutrient providers, pollinators, etc. Due to their high diversity, a wide range of these insect communities require specific habitat conditions, making them highly useful bioindicators for determining habitat quality. As general clear-cuts remove the overall availability of wood on the stand, including dead wood, they remove overall availability for insects to inhabit the stand, impacting the abundance and diversity of insects in the process. Practice with wood retention and residual, such as partial harvest practices, have not only found higher values of insect abundance and diversity, but also found that these practices are able to maintain a part of the community from before the harvest. Now, in similarity to the insect community, the vertebrate community conditions also respond to the impacts of their environment, often visible in the availability of dead wood. Forest-dwelling invertebrates show high precision in choice of dead wood, favoring a wide variation in wood quality and wood quantity, which are highly dependable on the forest conditions after harvest. Furthermore, as vertebrates more commonly rank higher in the trophic food chain, their dependency can and has been linked to the quality and quantity of lower communities, such as prey invertebrates or seed-bearing trees and shrubs. Here again, partial harvest treatments have the ability to retain crucial and beneficial traits to the vertebrate community, such as nesting availability, food resources, shelter, which through the severe impacts of clear cutting are otherwise scarce or unavailable. So, for the identification, each community has their benefits, but also its constraints. Plants are not mobile, so they are very dependent on their direct surrounding. Insects are extremely diverse, but very competitive and hard to identify. Vertebrates are extremely mobile and often require a larger habitat to monitor their selection. So, to tackle this dilemma, which approach is the better one, we are using not just one, but all 3 taxa as a bioindication approach, with the different community sensitivities to various measurable conditions such as spatial scale, coarse woody debris, wind throw, tree retention, etc. We assume and hope that the multi-textile approach has the potential to detect various direct and indirect fluctuations linked to the changes in the harvesting approach, and therefore creating an accurate representation of the ecological condition on each experimental unit. So for our approach, in order to find out which treatments are more suited for the conservation of the ecological integrity, we decided that each chapter covers one community response. In total, 3 communities are observed and compared over partial harvest with clear-cut and untreated management, and combined, these responses all come together as one bioindication approach. In this evaluation, our general motion suggests that the partial harvest treatments provide an overall more suitable condition to the boreal forest biodiversity. This due to various reasons such as higher variation in microhabitats, distinct higher conservation of crucial elements, and the reduced harvest severity of the practice itself. Now, in order to achieve the evaluation, we begin with the vegetational responses in the understory. In the first chapter, we focused on the response of understory vegetation to modern silvicultural methods on boreal forests. Hereby, we suggest that intermediate harvest intensity as tree removal and harvest severity linked with partial harvest increases the diversity of the boreal forest floor vegetation. This due to the conservation of mature biotic and abiotic traits. In the second hypothesis, we state that in regard to clear cut, the presence of adjacent residual stands accelerate the community maturation and increases the diversity in the forest floor vegetation. This due to close reach supply of seeds and near mature environmental conditions made available by the continuing presence of mature vegetation through residual. In the second chapter, we highlight the silvicultural impacts on invertebrates regarding the effects of retention and harvest method on the invertebrate communities. In this subject, we suggest that the conservation of dead wood, forest litter, and woody debris in combination with harvest intensity have an influence on the invertebrate community. Furthermore, in the effects created by partial harvest, we propose that the invertebrate diversity and abundance are positively correlated to the volume of windthrow and the intermediate harvest intensity. Thirdly, we suggest that due to the intermediate harvest intensity, higher harvest complexity, and higher spatial heterogeneity, the invertebrate community composition of the stand will have more similarities to the untreated stands than those with lower or no values of these traits. Coming to the final chapter, where we evaluate the post-harvest vertebrate species richness and diversity 20 years after impacts, as well as with the other species communities, low and moderate harvest intensities, such as tree removal and harvest severity, are positively correlated to the long-term species richness and diversity values of the vertebrate communities. In relation to the trophic levels, we go even further into suggesting that the high diversity and abundance of the understory vegetation and invertebrate communities have an impact and are positively correlated to the vertebrates, that is the bird and the small mammals, community diversity in boreal forests. In further environmental conditions, the retention of green trees and higher volumes of dead woods, standing or lying, in partial harvest treatments increase the vertebrate diversity more than the untreated and clear-cutted treatments. So, how are we going to find this out? Well, coming to my study location, the study area of the experimental sites are located within the North Shore and Mont-Valais region of Quebec. The region's climate is tied by a subhumid subpolar condition with annual temperatures between 3 and -2 degrees and a short vegetational growing season. The landscape is a typically forested hilly landscape interwoven with lakes, swamps, rivers, and rocky outcrops. Overall, the environment remains stable with a low frequency of heavy disturbances such as forest fires and a moderate to high frequency of minor disturbances such as storms or insect outbreaks. For the study design, the experimental sites and units for the project were established in 2003 by the Canadian Forest Service. This for the purpose of uncovering more information about the silvicultural designs in black spruce forests. In total, 6 sites were selected, containing each 6 treatment units. Of this selection, 3 of these sites resided in immature and 3 in mature. Each experimental unit, as you can see here, covered 3 hectares, buffered by a 30-meter residual patch to avoid influences from the surrounding landscape. This study focuses on each and all of these units for the monitoring of the community responses in our project. So within each site, there are 6 different treatments. 5 of these were mechanical and contained 3 distinct partial harvest treatments, which are the mini strip, the distance selection, and the close selection. Then we have 1 more intensive seed tree and 1 very intensive clear cut, leaving the 6th one with the control group. Besides for the spatial differences in spatial patterns, as you can see in the partial harvest, there is also a difference in residual, retention, and harvest type and complexity of the harvest treatments. Beside that, also the intensity of the harvest ranges between 0 for control, 50 for partial harvest, 75 for seed tree, and 100% for traditional clear cut. Now, for the observations on the units, a standard permanent sampling plot is used in each and all of the experimental units over all the chapters. The sampling plot is located as the core of the unit, ranging 600 square meters, 60 on 10. This to obtain the information of all the structural and spatial heterogeneity in each treatment. In total, 1 was placed in each unit, so 6 per site and 36 overall. In addition, 2 transect lines follow the length axis of the permanent sampling plot at 5-meter distance, with each 21 circular microplots placed at 3-meter distance between each core point, totaling at 42 plots per unit and 250 plots per site, with each a radius of 1.13 meters for each microplot. The sampling interval is done on a yearly basis and collects 10 characteristics such as density, age composition, and environmental characteristics such as precipitation, temperature, light intensity, etc. So for the collection of the understory vegetation, we use the microplots present at each unit. The collected data included species count, coverage percentage, species condition, and collected these data before the harvesting was done, during the harvest year itself, and 1, 10, and 20 years after the harvest, all done between June and August. For the understory vegetation, additional variables were also collected specifically for the explanation of the vegetation community responses, which are linked to the understory vegetation, such as the humus profile, the mounting soil, trail impacts, scarification, solar radiation, etc. Now, for the monitoring of the invertebrate community responses, we collect and identify insects over each separate unit via pitfall, crossvine, and soil samples. For the pitfall traps, 22 are placed within each unit, 14 of which in the core and 8 of which on the edge, totaling at 132 per site and 792 overall. In the core, the pitfalls are placed at the center of every 3rd existing microplot, as you can see in the orange balls. This is done for both sides of the permanent sampling plot, coming into a total of 14. And at the edge, 8 are placed in a square formation, with 1 at the center of the corner and 1 at the center of the edge, ranging a total of 47.5 meters between the nearest neighbor. For the crossvine traps, 5 were placed every 30 meters across the line transect, which runs through the center of the unit, parallel with the permanent sampling plot, totaling at 30 traps per site and 180 in total. Finally, the soil samples were taken inside the core of the unit for the observation of the microorganisms, which is done by my colleague Sang Yun, and these are additionally analyzed for soil invertebrate identification. So, for the trapping. The trapping containers of the pit and crossvine traps contain a conservation liquid existing out of 75 to 85% ethanol and 15 to 25% formalin with distilled water. This to conserve the baited insects without damaging the tissue. All trapping will be done on a yearly basis every June to August, and each trapping session consists of 30 days, upon which the traps are emptied for identification and inventory and reactivated for another session, repeating this procedure until the end of the trapping season. The additional sampling was also done for the invertebrate, for explaining the invertebrate community responses, which are typically linked to deadwood traits, such as volume, species, standing or lying deadwood, sun-exposed deadwood, etc. Now, due to the high diversity of the insect community and the often difficulty to determine the invertebrates, all trapped and collected insects are additionally sampled and analyzed via DNA sequencing. This is done according to the DNA EASY protocol for invertebrates, after which they are sent to the Centre d'Expertise et Services de Génome du Québec for further identification. For the data collection of the vertebrate community, we focused on two different taxonomical groups, namely the birds and the small mammals. For the birds, the data is collected via two methods. First, the field observations, which is done by two field observers performing distance sampling on a line transect which spans the whole length of the experimental unit, as you can see here. Each unit is monitored 4 times per year, bringing a total of 144 monitoring sessions over all the sites. Additionally, bioacoustics monitors are placed at the center of each unit, which record the unit for 2 minutes every 5 hours after sunrise between the months of May and July, giving a total of 546 recording hours per year over all the sites. So for the small mammal community monitoring, we applied a non-invasive observation method which determines the community of the small mammals via the DNA analysis of the hair samples. This will be done via tubes containing double-sided tape inside, which collects the hair upon the run-through of the animal. The sampling grid within each unit is built up from 5 sampling tubes in a square formation, with 1 at each of the corner of the square and 1 at the center of the square. In total, 180 tubes are activated each year from May until July, starting 2023, after which they are recollected in July for DNA analysis. The DNA analysis is again done with the same protocol for the DNA EASY of Kayagan, after which they again are sent to the Centre d'Expertise et Services de Génome du Québec for further identification. So, for the statistical method, the analytical method to approach and process the collected data is performed through 3 specific steps. First, all the collected community response data are analyzed for each separate unit, followed by the site age classes, and then the overall. When this is done, all the community responses can be compared over the harvest traits, finding the trait differences among the treatments and their effects on the responding communities, ending with the final determination of the main drivers of the community change and their respective quantities over the treatments, revealing the actual differences between the harvest types. For example, if we find that moisture is an influence to the diversity of the understory vegetation, we can find out their quantities of moisture between the partial harvest and the clear cuts. Now, in this study, all the collected traits directly serve as a response data in the statistical analysis. The fixed factors include all the implied experimental designs executed on each stand and the selected stand age. Furthermore, the explanatory factors are all the collected variables observed during the sampling sessions are extracted from online governmental databases. And the unit and site serve as random factors which influence the response data. So, even though the statistical analysis is performed to look alike over all the 3 chapters, as I explained before, but each chapter tries to highlight a different analysis form. For the community analysis, the presence, absence, Margolef's index, alpha diversity, Shannon-Wiener indexes are used to analyze the community richness and diversity responses, of which the most optimal is selected from the data outcome. Further, the principal component and coordinate analysis are further used for the analysis of the community composition of each treatment type. In the following step, we use the Bray-Curtis similarity of variance, the multivariate analysis of variance, and the permutational analysis of variance to analyze the differences among the community responses to the various treatments. Finally, finishing with the influence analysis of all the traits separately, where we analyze and model which explanatory traits best explain the community variation with the generalized linear modeling or generalized estimated equation in RStudio. So, for the originality of this project, we can list several reasons. When we look at the harvest design, the condition in which we study the community in partial harvest experiments on black spruce forests in 3 modern experimental partial harvest designs gives this study already a unique approach. On top of that, we use new approaches to determine the biodiversity or ecological state of the forest ecosystem by the use of the multi-taxal approach, which gives us a unique opportunity to test a more advanced approach of bioindication. Furthermore, the DNA analysis further fortifies a well-established bioindication method for determining the ecological conditions of the forest. So, in summary, this study contributes by providing new information on the community responses and partial harvest approaches and their benefits compared to the traditional and unmanaged treatments. It provides new insights on the uses of bioindication with new promising indication methods, resulting in the provision of essential information useful for the creation of ecologically sustainable management on an ever-changing environment that is in dire need of sustainability. Thank you very much. I would like to thank all the people that made the creation of my PhD possible and helped me to get to the point where I am now.
Thank you so much, Ruben. Now we are going to realize the question for the public, and then I invite to the people in the room of Amos. If you have questions, you can ask to the student, and after the people that are online. Gabriel, yep. Somebody in Amos?
Hi, Ruben. Congratulations on your presentation. It was very, very interesting. You have a nice project and a lot of work ahead of you. I was wondering, you said that these are permanent study plots.
Yep.
How long they've been there for 20 years, or are you like planning to put new ones?
They have been there for 20 years, and they've also been used in other studies before me, such as my supervisor himself used these plots as well for his research. So I am taking over these permanent sampling plots as well in my research.
Okay, great. And do you think that there will be maybe an effect of previous studies on your results, or do you think that it won't? Will you account for that, or yeah, that's actually my question.
I can look into that. I have not thought about that directly, but I can look into that. Yeah, thank you.
Okay, great. Great work.
Thank you.
Merci. Plus de personnes, Amos?
Oh, yeah, I have a question that since I already been to that field place, I was wondering, like, have you concerned about the size of the experimental unit, which is 3 hectares with 6 meter buffer zone, because it may be a little bit smaller than usual clear cutting size in general in Canada. So have you concerned those differences among those the clear cutting size or other partial harvest area on biodiversity?
Can you repeat the question? Sorry, I didn't hear it well.
Have you concerned the size of the experimental unit, which is 3 hectares with 6 meter buffer zone, because it may impact on biodiversity consequences?
Yes. So the experimental sites by itself, the 3 hectares, were already established beforehand. And so this is not something I can change. I cannot expand this because the research that I'm doing is a long-term research study. So I will be looking into this, but over a larger range. That's why there are constraints for example, the larger vertebrates. We have to exclude, for example, the larger ungulates because the observation is not possible. And that's why we chose as well for species that are not extremely mobile and that we can still observe in this area.
Okay, thank you.
Okay. Plus de personnes, Amos, on demande s'il y a des questions à distance. Okay. Personne à distance?
Yeah, I have one question.
Okay.
Ruben, for the micro mammals, the small mammals, will you use baits in the traps?
No, I don't think, no. In other research that I've already read about this, they use what we are also trying to plan for the small mammals is actually try to find common transportation routes that have already been used, where you can see that the small mammals use these areas or these traveling locations either way. And if we place the tubes there, it might have a small impact, but not too big to make them avoid this range, so they can still run through it. but I will not lure them into debates, no.
Okay, thank you.
Plus de questions pour l'étudiant?
Yes, I have a question related to the one from Sengyun. You say that you focus on mammals with small range or vertebrates with small range, but I have some concern regarding the range, the home range of birds, because some of them are able to migrate, and
Yes.
a 1 hectare or 3 hectares study area. So how can you be sure that these birds spend times in this location? And
Yeah.
especially with only 2 minutes of recording.
Yeah, so we are going out from the bird calls that are being done during the sunrise, where they have been most active during this their territorial display. And we are not going to like tag or follow a bird. No, we are monitoring the selection of the birds at the site itself. So we are also not going to follow the animals outside of the site. We are mainly focusing on the animals that are inside of the site itself. Even if these are, for example, have a very high migration routes, such as birds, we can still observe the differences in their selection. For example, birds that need shelter or nesting sites will not go to clear-cut sites or agricultural sites when they require trees or dead wood to make nesting. So we can find out if these trees select these areas, that they prefer the area over the other.
Okay.
It's more of a selection preference than actually monitoring the animal and their direct movement behavior. Yeah.
Okay. Yeah, but some birds may sing more louder than some others. So how can you be sure by recording that this bird finds itself in the study site and not in the buffer zone, for example, or in the surrounding habitats?
The range of the bioacoustic itself is not that far that it reaches until the end of the 3 hectares. So we already have the limit of the bioacoustic that makes sure that it actually mainly can observe the bird calls that are done within the core of the unit. And additionally, we have the distance sampling, which is done by the 2 observers that further confirm the animals of actually being there or not.
Yeah, okay. Well, if you think so, that's good. But I think because birds can sing really loudly, so yeah, you might find some record of the birds that are finding themselves in the buffer zone. So
yeah, of course there are going to be some variables that extend very, very far for some bird species, but if we do not encounter them by the distance sampling itself, that's already less viable information. So we have a 2-step actually confirmation of the bird species present within the site. So we can confirm if the species are both encountered by the distance sampling and found on the bioacoustic monitor, we can be certain that the bird is there. Well, if it's just for one of the 2, okay, our estimation is less exact, but that happens with any study.
Yeah. Okay. Good luck.
Yeah, thank you.
Parfait. Je pense qu'on n'a pas plus de temps pour plus de questions parce qu'il faut continuer. Alors, merci à tout le monde d'avoir accompagné à Ruben aujourd'hui. Si vous avez plus de questions pour lui, vous pourrez le contacter. Et on vous invite à partir et à quitter la salle parce qu'on va commencer le tour de questions du comité.